So in lab, you're going to have to do primary stain and then a counter stain. And so it's going to be a little bit different than the gram stain. Because with the gram stain, you were able to use that double rods going across. This time, you need to use the tripod because you're going to have to you're going to make your smears just like you did last time, but you're going to have to put the slides there, and you're going to have your flame underneath them to dry them and percolate them for, I think it's like 5 minutes. Yeah, because you're going to place it over the tripod, which is over the enamel staining dish, so that way if you do drip, it can catch it. And you're going to be putting a dry piece of paper towel over your slide, dripping the stain, your malachite green over it, and you need to keep that wet for like 5 minutes. And then we'll take it off. So that's the big thing you're doing today. You're doing an endo stain for 2 different things, two different strains. So hopefully you'll see these nice, pretty malachite green, showing you where you have the endospores versus the vegetative cells. Okay? Does that make sense? Are there any questions? I mean, because everything else should be straightforward or similar to gram staining. Wash it off, add counter stain, wait, wash it off. Questions? Are there any questions in general? Besides that reminder, do you have a test tomorrow?
I just had a question. How, like, the green on that slide, like, how green is that to you? It's a weird question. I think I'm a little colorblind.
To me, on this slide, it looks a little purple. Or not purple, sorry, blue. It looks blue. But, well, some of them, like, this one looks blue, this one looks like green, this looks kind of green.
Okay.
So, to me, in this picture, it looks varying degrees of green. But then again, you have to remember, we're looking at pictures of pictures of pictures.
Yeah.
So, color distortion varies. I mean, when we were looking at the gram staining, I showed you pictures of the purple and the pink. Did it look the same purple and pink in your microscope? No, but you could say, yes, that is pink, and yes, that is purple. They aren't the same shade, but you know what they are. That makes sense?
Yeah.
Other questions? No? Okay, well then, let's finish up chapter 11. We'll go through it again and finish it up. Sound like a good place to review from or start back from? So, basic shapes of bacteria. So, coccus or cocci.
What's the difference? Is coccus one?
Coccus is
plural.
one, and then cocci is the plural. Yeah. So, bacillus, bacilli. And then you can also have other shapes, basically kind of ends up being a lot of times spirals and other intertwining things. Big difference here is this beryllium is very stiff, whereas it's very flexible. So having the difference between a stiff, rigid structure and a very flexible, fluid structure. These can actually, in reality, be stars or triangles or rectangles. So there's a wide variety in the pleomorphic. And the vibrio, something that doesn't very apparent in the picture, is that it's just got a slight curve. It's a rod with a slight curve, basically. Okay? So basic shapes. And then when we start talking about naming, some of this should be just incredibly intuitive. Neat. Di or diplo. 2. Okay, so Diplococcus, you have 2 round objects. Tetrads, you have 4. Okay. Strep is for the string, and then if it ends up being conglomeration, it's the staff. Okay, so that's kind of an important thing to remember because that carries through to the rod shaped. If it's string, it's streptobacilli versus streptococci. Okay, so very easy. Start learning some of those roots. Start making so it makes more sense. Saracen, got a nice little cube looking thing. Cuboidal cell arrangement. Makes sense? Okay. Bacillus, rod shaped, they can come singly, doubly, strep, as in strings, or palisade, which means they're just all folded in on each other. Binary fission. So, why are we looking at all this? Well, bacteria is the most diverse group of cellular microbes, and so it's kind of important to know what they do and how they're doing this. Binary fission. You replicate your DNA, start moving it to the different poles, you get that membrane cell wall forming, and then that cell wall completely forms, and now you have two daughter cells. So, with binary fission, two daughter cells, the mother cell is completely gone because it created the two daughter cells. In the budding, you retain that cell and you just kind of get that little projecting off. So what's another form of binary fission? Snapping. The snapping division. Okay, so where it just kind of pulls the part. And then, so you can do the binary fission. This is also binary fission. It's cutting it in two. You can do binary fission. You can do budding. Replicate, kind of shove some DNA and information into a bud. So you get that young bud, and you end up with your daughter cell. Okay. So, sounding familiar, making sense. Endospores. Okay, since we're going to be playing with these a lot today, so endospores. Remember, they are vegetative, means active. Endospores are when times are tough. You make an endospore. And to make an endospore, you actually duplicate your DNA, and then you have the two DNAs aligned, so it looks a lot like you're getting ready to replicate. What happens is the cytoplasmic membrane actually starts and just closes in on that second set of DNA. And so it creates the four spore, and then you get the cytoplasmic membrane comes and engulfs it again, so you get a double membrane, and the vegetative cell's DNA starts to degrade, so you only have the DNA inside this endospore, and then the cells start shoving calcium and diploconate acid inside, just kind of hardening in between those two membranes. And so this creates a cortex in between the two membranes of that endospore. And so this kind of helps the spore coat form. So you're actually getting that nice spore coat here, around them, so you're shoving stuff in the middle. You get that spore coat forming, you get, the endospore starts to mature. As it starts to mature and complete that spore coat, it's going to now have increased resistance to heat, chemicals. It's going to be incredibly hard to kill. We don't exactly know why. It's something they're studying. And the original vegetative cell starts kind of breaking apart, disappearing, so you're just left with that free endospore. Okay? So, vegetative cell to free endospore. Yeah?
What did you say was the acid in between the two layers?
They don't dry out, they're resistant to heat, they're resistant to chemicals, and we have two different things that make them, that which are, just a couple examples. And then pretty pictures of what they kind of look like, so you can just kind of see. You have these endospores, you have the rest of that vegetative cell. So it depends on the type of cell, if that endospore is going to form at the middle, or to one end, polar or centrally in that cell type. Okay, so here are your spores. Make sense? Ready to move on? Archaea. So, archaea, they're eukaryotic, prokaryotic, what are they? Prokaryotic, okay. So they are prokaryotic. They look kind of eukaryotic with some of their things, which is why they're not in the class bacteria. They have their own domain. So, wait a minute. If we're going for naming, what are they? Go domain,
Kingdom. Phylum.
kings play chess on fine green silk. Kingdom, phylum, class, order, family, genus, species. I just write the first letters. I can usually figure everything else out by that. And then I also have them in order. Okay, so, domain archaea. So the largest group of archaea is methanogens. They are obligate anaerobes. What does that mean?
Anaerobes, no oxygen.
No oxygen, okay. And so methanogens, what is that? You know, we're converting carbon dioxide, hydrogen gas, organic acids, we're converting all this into methane gas. Okay. What is methane? What's the chemical structure of methane? CH4. Okay. These little methanogens are great because they can convert some of the organic waste in ponds and lakes and oceans into methane. Archaea lack true peptoglycan in their cell walls. And a lot of times they get known as the extremophiles, but the mass majority of them don't actually live in extremes, but they are the ones that have that ability. I believe they were just saying they found some that were living in nuclear reactors kind of thing, so they're pretty special where they can survive. You know, because they were doing studies where they had radiated all their equipment, so it's supposed to be sterile, and then did tests to make sure they were sterile, and they're like, things are growing. Why are things growing after we irradiate them? So, they also have fun with some of there's been some extreme organisms they found. They have one they named Conan the bacterium, because they can survive. So yeah, they have fun naming things. These are great. They're thinking about biotech-wise. The use for them is sewage treatment plants. So, deeply branching and phototrophic. Deeply branching bacteria, it's because when scientists look at them, they think these organisms are probably very similar to the earliest bacteria. This is what they looked like way back when. You know, they live in habitats that are similar to those thought to exist on early Earth. They can live in hot, acidic, and anaerobic environments. They produce oxygen, being autotrophic, producing our food. So, but they can live in what was thought to be an early Earth atmosphere. Okay, so we have the aquifex, which is considered to represent the earliest branch of bacteria. Then Coppus, the outer membrane is similar to gram-negative, but stains gram-positive, so just some little goofy things with that. And then prototrophic, what does that mean?
Need light.
Phototrophic, sorry.
Need light.
Light. So, phototrophic bacteria, they contain photosynthetic lamella. So remember, we're talking about prokaryotes. So this means they don't have membrane-bound organelles. They're autotrophic. And they get divided into 5 basic groups. You get your blue-green bacteria, the cyanobacteria. Picture. And so you can see they're just pretty, the blue-green. Their big job is nitrogen fixers. So one of the issues we have is nitrogen in the atmosphere is not readily available. So in the atmosphere we have N2. So if you remember chemistry, that means N triple bonded to N. That triple bond is hard to break. And so for it to be readily accessible for like us to use or other plants, we need it in a more friendly form like NH3. And so nitrogen fixers are capable of breaking this nitrogen bond. So they convert N2 into NH3. So they take unusable nitrogen, free nitrogen, and convert it into a usable form. We also can get green sulfur bacteria, non-sulfur bacteria, purple sulfur bacteria, and non-sulfur bacteria. So, cyanobacteria, the sulfur, and you can, the purple sulfur, you can see the little sulfur deposits in it. It's a little yellow. I bet they smell really lovely. What kind of microscope are we using for this?
Oh, it's over here.
Okay, one of the other ways they really start classifying bacteria is based on the DNA and the concentration of C's and G's. Okay, so DNA. DNA is what? What are the bases of DNA? ATCG, right? 4 basic bases of DNA. A binds with
T.
Okay. An A to a T is a double bond. A C to a G is a triple. So you might see them like that as well. Organisms that have a high CG content tend to be more thermally stable. They have more bonds. They can handle more heat. Ones that have more ATs are less stable. Or, they just can't handle some of those extreme temperatures because they don't have as many of the triple bonds. And so it's one of those things where you can read across one string of DNA and you can count up how many C's or how many ATs, and you know it's just going to be doubled because every G binds with a C, every C binds with a G, T, yeah. So it's a nice way to split things up going up. Are we low GC concentration or high GC concentration? How likely is this DNA to come apart, basically? So that's one of the ways they have separated some of the bacteria into, you'll see in your book, low GC or high GC. Okay. An example of low GC content. And so they can be facilitated or obligate anaerobes. They lack cell walls. These are the smallest free living cells. And these have importance in industry, environment, healthcare, just kind of learning about them, knowing what they're doing so we know how we can use them and how we can kill them. Okay. Also, some of the low GC bacteria, like Bacillus, it's common in soil bacteria, it's common in soil. Lystera, it often contaminates the milk, so it's one of those things you want to know about, so you know if you have contaminated milk or meat products. Lactobacillus, it can sometimes grow in the body, but it rarely causes diseases. Or you start getting like Streptococcus or the Enterococcus, and those cause a number of diseases. Or Staphylococcus, it's one of the most common inhabitants of humans, it's on our hands. Okay. Yeah, so low CG. There's your lovely list. So sometimes humans, diseases, rarely diseases, problems. Make sense? Okay. When we talk about BT and BT toxin, does anyone know anything about BT toxin? No? Never heard of it? Okay, so these crystalline products, this BT toxin that is created, it's called BT because it comes from the Bacillus thuringiensis. And so since the organism, BT, creates it, it's a BT toxin. But what we found with this BT toxin agriculturally is that it kills caterpillars that ingest it. And so a lot of organic farming will go along and you just spray this bacteria, they'll create BT, and then caterpillars will eat it and they'll die, so then you don't have to put pesticides on it because the bacteria will do it, and that's considered organic. Industry-wise, they've taken this BT toxin gene and they've put it into the crop directly. And we're not going to spend time spreading this bacteria on everything. We're just going to insert that gene into the cotton or corn. They have BT corn and BT cotton are the two big ones. And we're going to let the crop produce this, and then it's not going to be eaten by the caterpillars because they'll start to and then they'll die. So that's kind of the different takes on how you can use the toxin or how you can use the organism, either your organic farming or your biotech GMO farming. Make sense? Same outcome. Caterpillars die, higher crop yields, but are you going to spread the bacteria on top, or are you going to just incorporate that one single gene? So these are kind of all examples of high CG. So this is the mycococcal bacterium. And then this one, which is the high CG, it's gram-positive. One thing you can see here is you can clearly see the spores kind of coming off, being kicked off of it. And it's thought this might cause disease, it's Nascidomyces. So, form branching filaments, they resemble fungi. Yeah.
The BT toxin was what? Which one of them?
BT toxins what?
Like low or high?
Well, because I believe that's a high BT, because it comes from Bacillus thuringiensis.
I didn't know if you said it or not.
Well, I didn't say it. Well, Bacillus is low, so that's a soil bacterium. I didn't have that one specifically listed.
Okay. Thank you.
Okay. So, little nodules on pea roots. If you've ever wondered why wine grapes, like if you go through the Napa Valley, you see all that mustard out, right? The reason is because of having little nodules like this on this pea root. So these are rhizomes. So mustard, what they'll do is they will facilitate. So the rhizomes are little, basically it's a understanding between the plant and microorganism going, here, I will create this nice, safe place for you to grow, and you fix nitrogen for me so that I can grow. Okay. So it's the rhizobium nitrogen fixing. And so this is just one of the, I like the little picture because, I mean, that's one of the ways that wineries don't have to put nitrogen into the soil. They can let the plants do it and get that nice nitrogen boost. And then, so they're gram-negative protobacteria, the rhizobium. And then another interesting plant one is the agrobacterium. You guys ever seen these? They're called plant galls. You ever seen these on any of your trees or? Okay. So they're caused by agrobacterium, and what they do is they infect the tree, and they basically tell the tree grow. And so this tree grows, and basically just starts producing food for the virus, for the bacterium. Okay, and so they proliferate. You have undifferentiated cells, the gall cells, and that gives all the nutrients to the bacteria. And so plant biologists have actually taken this, and because agrobacterium is really great at infecting plants, and so they've gone in, changed the DNA a little bit, added plasmids, there's a special plasmid that is part of the agrobacterium, and they now can transform plants with whatever they want using a bacterium that's just going to make them grow. Are there any questions? Well, that's chapter 11, so we went back through it. Is there any other basic questions, things you want to cover? The test is, I think it's 110 points. I think it's 95 are multiple choice or true false, 15 are short answer. And by short answer, I mean like one word. It's a fill in the blank short answer.
Will we be doing anything tomorrow besides the test?
Lab.
The lab? There won't be a lecture?
No lecture. All lab.